Aseptic technique is the transferring or moving of microorganisms from one growth medium to another. Aseptic means without contamination. Literally, a means without and septic means contamination. By using aseptic technique, you can only grow the microorganism you wanted to study without the presence of unwanted microorganisms or contaminants. The first is the hand washing technique. Hand washing must be done before and after all lab activities, and it should be done properly in order to ensure that contamination is prevented in the laboratory. After soaking your hand, apply soap. Rub hands palm to palm. Rub the back of each hand using the other palm with fingers interlaced. Then rub palm to palm with fingers interlaced. Rub the back of your fingers to opposing palms. Rub each thumb clasped in opposite hand in a rotational movement. Rub tips of fingers on opposite palm in a circular motion. Rinse your hands with water. And wipe hands thoroughly with a clean towel. Use the towel to turn the faucet off. The second is disinfection. Before and after using or working on your working area, disinfection must be done. After hand washing, you can now wear your non-sterile gloves, but always check for holes and rips. Do the same thing for the other glove. Wear safety goggles when handling liquid disinfecting chemicals to protect your eyes. Grab your spray bottle and your clean cloth and spray the surface of your working area. Wipe the surface and equipments using a clean cloth. Fold the cloth over for a new or clean surface area. Now that you're done, properly dispose the gloves in the prescribed bin. There are two options in sterilizing, and one is by autoclaving. First step is to wash all glassware with soap and water. Just assume that the covers are petri dish and the spray bottles are test tubes. Dry all the materials using a tissue or a clean cloth. After washing the glasswares, cover test tubes with a cotton plug and cover the cotton plug with foil to avoid contamination during sterilization. Envelope petri dishes and other glasswares with paper to avoid breakage during sterilization. Let's assume that the pot is an autoclave. Fill the autoclave with sterile water up to 1/2 inch below the pan. Next is place the glassware in the autoclave on top of the wire basket. This is how I place the glasswares on the assuming wire rack. So I place the assuming test tubes on a bowl because the wire rack's gap is too big. Next is to cover the autoclave and make sure that the bolts are tightly closed. Turn the burner on low heat and sterilize at 121 degrees Celsius at 50 pounds pressure for 25 to 30 minutes. At the end of the sterilizing period, off the heat source and allow the autoclave to cool down and release the pressure to zero. Release the bolts and slowly open the cover. And now you may remove the sterilized glasswares. And the second option is sterilization by flaming. There are rules to follow when doing this technique and the rules are on the screen. The first thing to do is to flame your loop. Second is to remove the plug and flame the tube. Transfer from broth to plate. Flame the tube once more and cover it with the cotton plug. And lastly, flame your loop again for sterilization. The next technique is the inoculation technique. Inoculation is the transferring of an inoculum into a medium where they can grow and multiply. For broth to broth, flame the loop. Next is to remove the cotton plug and flame the tube. Dip the loop, flame the tube, and cover. Next is to open the second test tube and flame it. Dip the loop once more, flame the tube and cover. For sterilization, flame the loop. For the solid to solid, flame your needle. Gently poke the needle over a colony. Open the test tube and flame. Gently poke the needle 3 times. And flame the tube once more and cover. For sterilization, flame your inoculating needle. The last but not the least, the solid to broth technique. First, flame the loop. Gently rub the loop over a single colony. Remove the cotton plug and flame the test tube. Dip the loop. Flame the tube once more and cover. Flame the loop to sterilize. And that's how you conduct the aseptic technique. You must go through hand washing, disinfecting, sterilizing, and the inoculation itself. I hope you learned something from me today. Bye!